Cell division is happening all the time in your body in order to keep you healthy and growing. Cells have to divide so that they don't get too large. If they never divided, they would just keep getting bigger, and then they would have issues with getting rid of all of the waste they have and taking in enough supplies, materials to keep them functioning. Cells also divide for reproduction purposes. Asexual reproduction occurs through two processes called binary fission and budding. And for both of these, the offspring are going to be identical to the original parent cell. So on the left here, you see an example of binary fission, and the cell is just splitting into two identical daughter cells. For budding, organisms that do this simply branch off a piece of themselves, and that is how they reproduce. For both of these examples, the offspring are going to be genetically identical to the parent. Cells also have to divide so that an organism can grow and just repair and fix cells that are damaged. In your body, 6 million red blood cells are produced every few seconds. And every time your cells are regenerating and growing and dividing, this is the process of mitosis. So you use the process of mitosis in order to make new cells to grow and to repair damaged cells. The cell cycle consists of a few different phases. So mitosis, this green part of the cell cycle, this is where the nucleus is dividing in the cell. Cytokinesis quickly follows mitosis, and that is where the cytoplasm is splitting and actually dividing a cell completely into two. But what has to happen here before a cell can divide? What does a cell have to do to prepare for cell division? Well, a cell must grow. It has to get better, bigger, and it has to just make sure it's ready for division. And then it also has to make a copy of all of its DNA so that when it divides, each cell is getting a copy of the DNA so it can be complete. A cell must have space to divide. If it doesn't have space, it won't divide. This is called contact inhibition. The biggest part of the cell cycle is called interphase. Within interphase, there are three different phases. We have G1, which is cell growth, S phase, which is for synthesis, or where the DNA gets copied, and then G2 is another phase of cell growth. There are also checkpoints along the way in the cell cycle. The cells can only move on to the next step if they are ready. So at the end of G1, there's a G1 checkpoint. The cell will only move into S phase and copy its DNA if it has gotten big enough. And then now it has a copy of the DNA, and so it can grow more in G2. At the end of G2, there's another checkpoint so that the cell can make sure it's ready to divide and that everything is looking okay. If the checkpoints are not working, then this is something that leads to uncontrolled cell growth, and that's the definition of cancer. When cells go crazy and they can't check themselves and fix themselves, that leads to cancer. So again, in summary, the cell cycle is made up of a big phase called interphase. That's where a cell spends most of its time, and that consists of G1 for growth, S for copying the DNA, and G2, which is also for growth. After that happens, a cell enters M phase, which is where the cell is actually dividing. M phase first goes into mitosis, which is specifically the division of the nucleus and the DNA that's in the nucleus. And then cytoplasm is just the division of the cytoplasm and all of the other parts that are out in the cytoplasm. What form is DNA in during S phase of interphase? So DNA comes in two forms, the long stringy form called chromatin and chromosome form, which is when it's really tightly compacted into that X shape. So in interphase, DNA has to be copied, which means enzymes have to be able to get in there and read the DNA in order to make a copy of it. So which of these forms do you think enzymes would be more likely to get in there and do their job? Well, that answer would be chromatin. When it's more relaxed and long and all stretched out, that is when the enzymes can get in there and actually make a copy of it, which is DNA replication. So in interphase, DNA is in the chromatin form. Make sure you know that cells actually do spend most of their time in interphase. So this is just when the DNA is long and stretched out, which means it can be getting used. And since it carries the directions to make proteins, that's important. Enzymes have to be able to get in there to transcribe the DNA so it can be used to make proteins to keep you alive. After interphase is complete and the cell has successfully passed checkpoint for G2, then the cell is ready to divide. When this happens, the DNA has to condense then into that chromosome form, which is so important. It compacts into that nice X shape so that it's more movable. And when the cell is moving it around during the cell division process, the DNA is then less likely to get all knotted up and jumbled up and just become this big intertwined mess. So now that the DNA is in chromosome form, the cell is ready to actually divide in mitosis. So mitosis is made up of 4 phases. You have prophase, anaphase, metaphase, and telophase. Inside of each of these phases, there is something specific that's happening in the cell as it's dividing and moving from one cell into 2 cells. So for prophase, if you think of the word prepare to remember what's happening here, the cell's just preparing to divide. So the chromatin has condensed into chromosomes, and these little features called centrioles appear, which is what actually anchors the spindle fibers. And the spindle fibers are these little strings that can attach to the chromosomes and pull them around to where they need to go. The nucleus also is going to temporarily go away at this stage so that the chromosomes are free to move to where they need to go. Metaphase happens next. If you think middle, that'll help you remember that the chromosomes at this point in mitosis are lining up in the middle of the cell. Once the chromosomes are in the middle of the cell, anaphase happens, and this is where the chromosomes are getting pulled apart. So since a chromosome is getting split apart, that actually means that the chromatids, which would be half of a chromosome, is getting pulled to a side of the cell. So the chromatids are getting pulled to opposite ends of the cell. Telophase is the final stage of mitosis, and if you think of the word 2, that'll help you remember that a cell is splitting into 2 during this part of mitosis. At the end of this, the nucleus will reform so that each of the resulting cells will have the DNA enclosed in a complete nucleus. After M phase, after mitosis specifically, that's when cytokinesis happens, and that's just division of the cytoplasm. So the gooey stuff that fills the cell and all the extra organelles and features that are floating around out there will get split so that the 2 cells that get made they're exactly the same. They have all of the DNA they need, they have all the parts they need to be functioning cells. Cytokinesis in plant cells is a little different. They just have an extra step, which involves a cell plate that will grow in the middle of the cells during telophase that will completely separate them and allow them to turn into 2 different cells. At the conclusion of mitosis, you will have 2 cells that are exactly the same. So the important thing to know is that all of the DNA is going to be the same in both of the cells, which is it's very important because if your skin cells are going through mitosis and making more skin cells, you need them to have the same DNA in them because that's your DNA. It's the DNA that's making you that codes for the proteins that are making you and keeping you alive. So every single cell that gets made in mitosis will end up with the exact same DNA in it. And that is what you need to know about the cell cycle and mitosis.